Good morning, everyone. So, during the outbreak of hepatitis of non-origin in children in 2022, at least 920 cases of non-AE hepatitis were reported. Using metagenomics methods, we and two other groups identified adeno-associated virus 2 in most of the samples we analyzed. We also found adenovirus and herpesvirus, particularly herpesvirus-6b. These viruses, adenovirus and herpesvirus, are herpesvirus of AAV2 replication. Our initial hypothesis is it was an immune-mediated response caused by co-infection of AAV2 with either adenovirus or herpesvirus. AAVs are commonly used as vectors in gene therapy, and hepatitis is one of the most common and severe side effects of gene therapy cases. Initial investigation into this case suggests that hepatotoxicity is an immune-mediated response, similarly to the one that we see during the hepatitis outbreak. So we wanted to analyze in more detail the similarities between these two types of hepatitis. So for this, we studied a case of hepatitis associated to a treatment of spinal muscular atrophy. So production of this treatment is made using three plasmids. You have PSMN, which had the transgene between two ITR from AAV, and you have two other plasmids that have AAV rep and cap genes and also adenovirus genes that allow AAV replication. For this, we use similar methods to the one we used to study the hepatitis outbreak. So if we use short-read metagenomic sequencing to do virus and plasmid identification and to study host transcriptome, and we use ONT to investigate the plasmid and viral genome structure. In order to look for similarities in the immune response, we compare cytokine modules in hepatitis cases, hepatitis B, and also in healthy controls. So you can see in the middle of the graph how the hepatitis gene therapy case clusters together with the 2022 outbreak, and they are different from both hepatitis B and healthy controls. Metagenomic analysis also shows us the presence of AAV, herpesvirus, and adenovirus, and we also found high coverage for two of the plasmids present in the treatment in the liver biopsy. Since these plasmids contain elements from both adenovirus and AAVs, these results likely indicate that the presence of the plasmids were there, but not a wild-type infection. During the 2022 outbreak, we used ONT to identify and analyze the viral structure of the genome of AAV2s, and what we found, we have really complex structures that show concatemerization, which is usually an evidence of AAV2 replication aided by either herpesvirus or adenovirus. In the hepatitis case, we also found complex structures for the plasmids present in the liver. So there are similarities between these two types of hepatitis. We have complex structures of AAV in the liver, and we also have similarities in the cytokine patterns for the two cases. Our hypothesis is this is a toxic effect, so a large amount of AAV DNA present in the liver is going to distort the cells and is going to put proteins for the immune system that they have seen before. However, it is also possible that T cells are raised somewhere else in the body against AAV proteins, and then these are initiating an immune response in the liver for this. So I would like to thank everybody that worked in this project, particularly Sarah, Christina, and Professor Judy Brewer, and I'm happy to take your questions. Thank you for your time.